do not have much of this kind, with some exceptions. But the first real cytoskeletal elements obviously came with the development of unicellular organisms. And there the mobility and the orientation also shape. Those were the challenges that stimulated this development and this evolution. Why wasn't it okay with those two cytoskeletal elements with more complex organisms? Obviously, because they're much more complex. And you have lots of challenges there apart from mobility and nutrients and other stresses. You have mechanical forces, cell interactions, polarity, mobility and orientation, and tissue integrity. Those are really major challenges. And when you look in evolution, the more complex the organism is getting, the more complex is the intermediate filament composition. So one could, of course, argue that we need this for mechanical purposes, but so that each and every tissue would have its specific mechanical support. But we have so many of these genes, and there's such a variation. If this I stole from our local community, so thanks to everybody who took these pictures. Sorry for no references with names, but this is just from our local community, lots of pictures of different types. And the spectrum of variability is obvious. It's quite amazing. And now these are obviously looking like threads. We know from high resolution images, they don't quite look like this. They're much thinner. It's a much more complex organization. But when people looked at them in the early phases of fluorescence microscopy and also electron microscopy, it sort of inspired the mechanical paradigm that these are for the intention of keeping tissues rigid, for keeping integrating tissue biomechanics and so forth. And obviously, like Bob Goldman pointed nicely out in his lecture, there is a lot to that. So they do have mechanical properties, and they do have very specific mechanical properties depending on the IF composition. But there's more to IFs than mechanics. And in the early 80s, the first observations came on post-translational modifications, out of which the observation by Lazaridis on pKa phosphorylation was followed by many different publications. Then Masaki Inagaki, who many of you know, observed that actually the assembly of intermediate filaments is dependent on phosphorylation. And now there were many similar stories around, and Bisher Omari was one of the first to actually show that there's a tight association between the filaments and different kinase systems, and there have been many follow-ups to that as well. It's been shown then later on that the phosphorylation is extremely complex. And you have many phosphorylation sites, and the post-translational modifications are not restricted to phosphorylation, but you have many other types of PTMs. And for example, Diana Toivola and Bisher Omari has a nice review on those, and that's not the only review on that matter. So this kind of extremely complex PTM patterns and systems, they're usually associated with signaling proteins. We know now, first of all, that the intermediate filaments are not leather, they're not rigid, except for those in the skin and nails and hair. which have then cysteine double bonds stabilizing them. But here we have a system which is actively turning over, and it's very dynamic, and this dynamism and these dynamic properties, they seem to relate to phosphorylation. And these proteins are usually kept at a very low phosphorylation stoichiometry, but the low stoichiometry really is not a result of them being statically at low phosphorylation levels, but they have an active turnover of phosphate. So there's an active kinase and an active phosphatase activity on each and every intermediate filament, and the composition of the kinases, also to some extent the composition of the phosphatases is variable. But this turnover is determining to quite a significant extent also the turnover that I was showing in the FRAP experiment. By the way, I have to go back to this shoe because it is one step back in memory lane. This is my shoe. I used this to illustrate the same purpose here, but those of you who were in Saint-Malo on the great meeting that Giselle Bon arranged on one of the most beautiful spots of all intermediate filaments in the history of all our nice meetings, you can see that there's the fortress of Saint-Malo in the background. Anyway, that was a parenthesis. Hello, Giselle, by the way. And so we have an extremely complex phosphorylation on these IFs, but routinely these phosphorylation motifs are also highly conserved, as shown here on vimentin. And you can see that there are many of these motifs that they're conserved all the way to frogs and fishes and so forth. And they are important for the assembly, they're important for the turnover. And if you mutate these, you can, in some cases, get really spectacular phenotypes, like in the sequence, which is at the far end of vimentin, where if you do a phosphomimetic and actually a phosphodeficient mutation with variable degree, you get very striking phenotypes indeed. And that's true of many of the other sites as well. But so the turnover of the protein is one, but then the interaction with signaling protein is another aspect where you need this PTM regulation. And we don't understand really much about this, but we begin to see the tip of the iceberg. This is a concept which we have developed in a number of different systems. It started with nestin, where we studied the interaction of nestin with CDK5, which is a very specific kinase involved in neural and muscle development and also muscle integrity, neural integrity. But I will not talk about that. I will focus entirely on vimentin in this presentation. And the idea is that you have a signaling molecule, just like in the early days, Bisher showed the interaction of a PKC epsilon with keratins. I will now discuss a number of signaling proteins interacting with IFs. And the idea is that you have a scaffolding system or a system which is a mechanical integrator of space, let's call it in that way, which has dynamic properties, which is all the time able to take in environmental cues, whether it would be from the environment, from the cell shape, from the neighboring cells or whatever. And this interaction will in turn also affect the process, and the process itself, because of the mechanical property of the system, will have a positive feedback into the signaling concept that we're talking about. Now, Bob Goldman mentioned the Vimentin knockout mouse as an example of an animal model which in the beginning was considered to have relatively mild phenotypes. But that's a good example, not the only one by any means, of an animal model which has been revealing its phenotypes only upon challenges. And now you're well aware of the different diseases that are associated with intermediate filaments. So in the Singapore group, which is led by Birgit Lane, there is this great intermediate filament disease site where you have lots and lots of different diseases which are associated with intermediate filaments. But characteristic to many of these is that they are revealed only upon some kind of stress, some kind of injury. And here you see a number of different examples. And my examples all come from wound healing. We know that keeping tissue integrity related to the first slide the second slide I was showing, where we have the complex structure of different intermediate filaments in different tissues, they are really supporting that integrity, not in terms of only mechanical properties, but maintaining that homeostasis and also shifting to other systems when you have a wound which needs attention, when you have healing which also needs attention, or in processes which are going wrong when you have chronic wounds or cancer. And cancer, as early pathologists were putting this, cancer can be considered in a way as a chronic wound, a wound that doesn't heal. So I'll be looking into Vimentin from all these aspects. And the project was stimulated by early observations on the Vimentin knockout mouse already around 2000, showing that there's a problem with wound healing. Fang Cheng, who was a postdoc in my group a few years ago, who is now at Sun Yat-sen University in Shenzhen in China, running a very successful lab. She really initiated this project, but now we have a number of team members that are doing different parts of this project. And I'll step a little bit back in time and give you the background. First of all, here's the observation, the physiological observation, where we could recapitulate what was observed in the early studies on the Vimentin knockout mouse. They do very poorly in terms of wound healing. And here's a burn wound, and you can see that this is highly compromised at 15 days. As you can see in the wild type case, you have almost complete healing, whereas in the knockout case, you have incomplete barrier formation and a situation that to some extent resembles the type of wounds that you may have in diabetic chronic wounds. I will not be presenting that study in any greater detail. I'll pick a few things from there, but then I'll try to analyze what is really behind this phenotype, because there are many things that are going wrong. One thing that is going wrong is that the immune cell systems are not working properly. And also the migration of immune cells, of leukocytes, T and B cells is not working properly. So here's an early cartoon of lymphocyte migration, where the lymphocyte is going and in the bloodstream. And when you have an injured site, they will stop, roll. There are signals on the surface of the endothelium telling them that there's something going on. And then they migrate into the tissue and start doing their job. I was working together with Sirpa Jalkanen, this is already now 15 years ago, on this, and they showed me videos that looked like this, where you have a transmigrating cell, which is there in the middle, and you have the cell border marked there in white at the end, at the tip of the cell. And you can see that this transmigrating cell, it's in the middle of this endothelial cell. You fool the system by putting a little bit TNF, and they think that there's a wound, and then they try to crawl through. And you could see that it was staying in place, and at the end, it was really crawling away, and it was crawling underneath the endothelial cell. So it had gone straight through the cell. And we used, of course, our favorite systems to show this. So this is a HUVEC cell labeled for Vimentin, and you can see that there's an immune cell that is smack in the middle of the HUVEC cell. And here's another where you have GFP tagged Vimentin, again in a HUVEC cell, and you have a transmigrating lymphocyte that is digging itself into the HUVEC cell. That was pretty cool. It was an interesting observation by itself, because it was sort of rewriting the history a little bit. This diapedesis or transmigration, transcellular migration was all observed already in the 60s, but it had been partly forgotten and partly it was considered controversial. But we, together with a number of other groups, could show that this is really happening, and nobody really understands why we need two different migration systems, but that's a different story. But along this publication, Nature Cell Biology required us to show what is the role of Vimentin, and actually we could see that Vimentin was required for the transmigration. So here you see cells that are adhering lymphocytes to the endothelial system, and when you have Vimentin absent in both the receiving and the migrating cells, it doesn't work. It doesn't work either when you have Vimentin absent in the migrating cells, and it doesn't work when you have Vimentin absent in the receiving cells. So taking a long story very short, so Vimentin is really required for the proper placement and positioning of integrins on the transmigrating population. And it's also required for the positioning of ICAMs in the receiving cells. And also in this publication, we could show that the endothelial integrity is highly compromised, and we had a follow-up study together with Cecilia Sahlgren's group, which was published in 2017, showing basically the same thing and linking this to notch function, so notch signaling. So I will not talk about notch, but basically from that study, what we could learn was that, and also subsequent studies, was that Vimentin is required for the proper cycling of PKC and integrin, which is enabling the positioning on the membrane of integrin. And Vimentin is also required for the ICAM positioning on the receiving endothelial cells by a mechanism which we don't understand. So continuing on the wound healing. So first of all, the immune system is not in good shape. This is obviously important. It's also important for the signaling. Let's go to the next topic, which is EMT signaling. And I'll just show you this briefly again, because this is published. We can see that, again, in the same wound healing situation, when you have the barrier formation being completed, and in the left-hand side, you have very poor barrier, we have also very little collagen. So the collagen deposition is compromised. Then because of this poor barrier formation, one could assume that also the EMT is compromised. So related to the collagen deposition, the study showed, and there has also been a number of other studies indicating that fibroblast growth in these conditions in the healing wounds is compromised, and we could show that this is actually due to a very low activity in terms of cell division, which again is due to a reduced ERK signaling. And so you can just see the knockout cells growing very poorly, and you can rescue that with increasing doses of vimentin, which also stimulates ERK activity. We still don't know exactly how this ERK activity is regulated, but I'll get back to the signaling partners towards the end of the presentation. Also, we could see that this is a cell system which is familiar to many of you. MCF10A cells undergo EMT when you stimulate them with TGF-beta, which you can see here in the upper row. When you transfect the cells with vimentin alone, nothing happens. If you stimulate them with TGF-beta, they undergo nice EMT, which is also shown by N-cadherin presence. The same happens in the normal cells when you stimulate with TGF-beta. Again, we have EMT, but when you have knockdown of vimentin, this doesn't work. So vimentin is really required for the EMT. And again, I will not take you through those data, but the signaling system here, going back to the signaling concept I presented in the beginning, is one where you have ERK interacting with slug. So the activation of ERK is necessary. The interaction of ERK with the activated ERK with slug is necessary. And keeping this all together is necessary for EMT to work out. So slug is one of the really early trigger of EMT. It's a transcription factor, and you have a number of events then following that. But this is the signaling concept again in this context. So when vimentin is compromised, and again here is the idea how this works, and that is another reason then why the wound healing works so poorly in these mice. Taking you through how far we have come so far, we have seen that fibroblast proliferation is poor. That results also in poor TGF-beta signaling, which in turn gives a poor activation of the keratinocytes, but also the keratinocytes require vimentin for their normal EMT. Due to the poor fibroblast proliferation, you have also poor collagen deposition. But you also need migration and reepithelialization. And so let's have a look on migration and what's happening there in that department. Now there has been a number of studies actually alluding to the possibility and also showing that vimentin or IFs are required for proper migration. And there was a nice study from the Danoser lab that was looking at the roles of vimentin in migrating cells and indicating that actually vimentin there has a role in terms of steering the key elements that are required for migration. From Sandrine Etienne Maneville's lab, there was in 2018 a study in astrocytes, which obviously have several different IF components, GFAP, vimentin, and nestin. But in that context, it was shown that the astrocytes don't behave normally, and that they have a problem with the migration. So coming back to fibroblasts, and we have seen the same thing in a number of different fibroblast systems. These are MEFs. And if you see the wound healing assay here, is indicating that they somehow look different. And they seem to be more messy towards the right-hand side. When you analyze this, you can see that there's really a loss of directionality. It's quite striking. And there's not so much a difference in speed, as you can see here on the right-hand side, but the directionality is really poor. It also occurs when you put the fibroblasts on a collagen surface. So you basically let them migrate on a collagen gel. And you can see that the wild types and the knockouts are behaving very differently. And actually, in the wild type situation, they participate in sort of structuring the extracellular matrix, whereas one could say that the knockouts are really making a mess, because you can see that the structure of the collagen gel is changing upon these migrating cells. If you analyze the tracks, you can see that it's very different. You can see that it's very straight in the wild type situation, and it's completely perturbed in the knockout situation. So these are single cell tracking. And we could see the same observation we could see with different cells, primary cells, and also with rat embryonic fibroblast knockout for vimentin. Now, we have published, and also other groups have published, the observations indicating that there's an interaction of vimentin with the actomyosin systems. So here from a study a few years ago already with Pekka Lappalainen's group, where we could see that vimentin is required for the formation of actin arch. And taking that summary of that study and again taking previous observations, we could have a concept, signaling concept that would look a little bit like this, where you have the a plectin vimentin complex that is regulating actin arcs. You also have an association of RhoA kinase, which in a not completely defined way regulates this, but it's definitely involved in regulating this. So now we can see that already we have the integrin is here. We have the actin arcs, which are regulating the flow of the cell. We have both of those components being defined. So what about focal adhesions? So in Sandrine's study, which I already mentioned, there was it was alluded that the focal adhesions indeed were perturbed. And if you look at these live cell imaging studies, where you have a number of different focal adhesions from fibroblast, you can perhaps see that there's a difference. So it's not so easy to see, but first of all, the size of the focal adhesions, they look different. And somehow they look messier in the case of the vimentin knockout fibroblast. Now, obviously, you have the possibility to analyze this in greater detail. And it turns out that indeed you have quite a mess there when it comes to the organization of the dynamic turnover of vimentin, of paxillin, which is the focal adhesion marker here. And you already see from these heat maps that they look very different. The direction is pointed out, and it's easy intuitively to tell that there's a difference, but you can also quantify that the focal alignment index is really poor in the vimentin knockout mice, the knockout fibroblasts, and then if you analyze the periodicity of the focal adhesion birth angles, there's also a difference, because the birth angles are formed with a certain periodicity, and that is quite obvious in the case of wild-type fibroblasts, but this is pretty much lost in the case of the knockout fibroblasts, again indicating that the focal adhesions are quite a mess in the knockout fibroblasts. The disassembly rate is higher in the knockout focal adhesions, and also when you do FRAP experiments, you have significantly shorter, faster recovery times in the knockout fibroblast, indicating a more rapid turnover, these are all pointing towards the same thing, that this vimentin is really required to maintain focal adhesion stability, and then the alignment results are indicating that vimentin is required to keep the alignment. Now, if we think about previous nice publications when it comes to the focal adhesion architecture, I think many of you are familiar with this beautiful study where you have a number of follow-ups that have been also published, where there was a super resolution characterization of the nanoscale architecture of the focal adhesion complexes. You can see that there is something missing here. There is no intermediate filament protein. So we wanted to check the nano architecture of the focal adhesions. Is vimentin really there? Because if it's actively steering the focal adhesions, it should be there. And actually, it turns out that it is really in the focal adhesions. So here showing the interaction of vimentin with focal adhesion kinase, which is obviously an important component of the focal adhesion. So vimentin sort of moves along with the focal adhesions, and you always have this dynamic interaction. So you have a certain site that is interacting with the focal adhesions. If you take super resolution images, like here with paxillin, which is obviously another important component, and you can follow the Z resolution at the right-hand side, you can see that vimentin is really going into the focal adhesions. And with the STED resolution, you cannot really distinguish what would be the layer where it's coming in the same way as we could show as we could see in this Nature publication. But this is what we're aiming for, to have a look where it could come in. There's another picture and showing basically the same interaction. And so this is our take on this. First of all, vimentin is, as I said, it's engaged in the determining the flow, but now it's also engaged in steering the focal adhesions. If vimentin is absent, they're really quite a mess. And as it turns out, into the focal adhesions, you have both full filaments coming in and also unilength filaments. Both of them are sort of stretching into the focal adhesions. So this again is our take on the same signaling chart, same signaling concept, and now what you could tell is that all taken together, and also previous studies taken together, why we need vimentin is really to maintain the organization of the migration and the components that are there. And they seem to be, in a way, orchestrating the different players in the orchestra. How that really goes in really molecular detail, we don't understand, but it's getting closer. For wound healing, you also need to regulate cell size. And one thing that has been obvious for many of us who have been studying the vimentin knockout mice is that they're very lean. They weigh less. And there has been also some publication also from Milos Pekni's lab. There was a small report about that last year. And when you look at these mice, they do have a very lean composition in terms of their fat tissue, but their tissues are also smaller. So if you look at kidney and heart, for example, they are really smaller. The length is not compromised, so that it's a really change in the leanness. As I mentioned, we already know that vimentin is involved in regulating cell division. Now, could it also be there to regulate cell size? It's attractive because cell size is also linked to autophagy, and there was a nice study from Beth Levine's lab in Science in 2012, linking vimentin to 14-3 functions regulating Becklin, which is an autophagy regulator. So it would be attractive to have this all together. So let's have a look. So first of all, vimentin fibroblasts are indeed smaller, and this is true regardless of whether you have them confluent as here, or whether you have them subconfluent, and it's true also for human knockout fibroblasts. If you look at cell volume analysis, especially with cell cycle block with thymidine, you can see that there's a quite significant difference. You can rescue that with vimentin. Also, cell protein levels are lower. You can see the same thing in many different cell types. Here we checked out with primary cells that are dendritic cells that are not adherent because they're in suspension by nature. And this is with Franziska Lautenschläger's lab. And we could see that both in the case when they're just without any stress, or when they're in a flow, which is stressing them or stretching them a little bit, there's a significant difference in the size. Now, if you take away the nutrients, this difference disappears. You can see it here. You have serum-starved cells, both the wild type and the knockouts are of the same size. Now, if you stimulate them with insulin or with nutrients, with fetal calf serum, you get a significant difference, pointing towards involvement of the mTOR pathway. And indeed, the mTOR pathway is really seriously affected when you don't have vimentin, shown here with the downstream signaling components there, just downstream targets indicating that you have reduced mTOR activity. We have been working on this for quite a while, and we have a monster manuscript that is has been already submitted a few times, and hopefully we get that published at some stage soon. And if you're a referee, please be kind to this. There's a lot of work behind this. Anyway, so the take-home message here is you have these different stimulations, amino acids, glucose, insulin, and we have here insulin in all these cases. So you sort of push the throttle all the time in this experiment, and then you have different types of gasoline, let's say in that way. And then you check the effects of different types of gasoline nutrients, and you can see that in all these cases, the downstream mTOR signaling is affected. Same thing with human HDF cells, human dermal fibroblasts, which we have now with CRISPR-Cas as a knockout system. We can see that also here, both the cell volume, cell area is reduced, and then we can see that the mTOR is significantly reduced. One critical thing there in mTOR signaling is that for mTOR to be activated, it needs to relocalize to the lysosomes. And because of time issues, we don't have time to dwell into the wonderful world of mTOR signaling, but it's extremely complicated. But one aspect is just the reorganization of mTOR to the lysosomes, and this seems to be really not working well in the absence of vimentin. So this is our take on this. Again, we have a signaling system where vimentin takes part. We don't dare to draw a cartoon which would be depicting the exact interaction and the exact molecular mechanism, but how it seems to be working, it's really favoring the localization of mTOR to the lysosome, which enables then activation, and which also is inhibiting autophagy. Which I didn't mention is that actually this P62 is really significantly affected in the absence of vimentin as an indicator of either stimulated or inhibited autophagy. Related to the fat observations, you can see that here is red oil staining of fat cells, and you can see that there's a really highly reduced amount of fat in the fat cells, and furthermore, the lipid droplets are really small. Werner Franke published in 1989, I believe, in JCB, a paper indicating that vimentin forms cages around lipid droplets. Since that, it has not been clear what is the relationship, but at least perilipin is really compromised in the vimentin knockout cells, and perilipin in turn is regulated by mTOR, so this could be an mTOR effect, but it could also be something else. It could be a structural issue, so we're looking into that. It could be also both. With all of this, I'm coming to an end. So obviously, we need to figure out what are the molecular players here. So far, we've been guessing, and now we have also gone into quite serious proteomics. So especially Leila Ratto in our lab is a proteomics expert and has really dwelled into this. And you can see that here's a volcano plot with the difference between knockout and wild type fibroblast. And there are a number of proteins that are coming up as hits, which are alluded to from the studies I was discussing before. So collagen, one of the collagens, ICAM, S100, which is a slug target, STAT2, which is a TGF-beta regulator, and vimentin obviously is a positive control, as it should be. And what we are also doing is nutrient independent and nutrient dependent interactomics, and we are looking. You can see that vimentin is interacting with lots and lots of partners, and there is a dependency, a nutrient dependency on this. And it looks like we have some targets that look very promising, both in terms of ribosomal functions, of mTOR functions, also focal adhesion functions, and autophagy. So let's hope for the best. I took you through a system which all relates to wound healing, where we have vimentin interacting with different signaling molecules. We have different systems stimulating this. This affects cell growth, EMT, cell migration, cell direction, ECM production, and also organization, protein synthesis and cell size, and vascularization, which I didn't talk so much about, but that was mentioned, and the notch studies mentioned in terms of regulating that. And so here's a philosophical view, or again, of the organization of IFs in different tissues, where one could maybe see a pattern that vimentin, along with a number of other IF proteins, would be either keeping systems in place or letting them go. So let's say if we have an epithelial injury, we have late keratins, which would be inhibitory. Vimentin would be stimulatory in terms of both proliferation, differentiation, migration, and invasion. Fibrosis, I didn't talk about, but also that, and reepithelialization. And depending on the situation, this can either lead to a normal physiological process or a pathological process. And similar systems you could identify in other organs as well. And if we take the EMT and MET, which is the reversal, one could say then that late and early keratins would be the opposite. Late keratins and vimentin would be the opposite in terms of letting it go. And if we take it to a signaling system, one could say that, or guess that there are signaling molecules that in one system they would interact with IF protein 1, and in an environment 1, and regulating process number 1. Now we switch system, we have a stimulus, we get another IF protein, we have another environment, we have other interactions, and we have other functions which are determined by the next IF protein. So much about physiology.